The goal of the following procedure is to rapidly and automatically quantify the changes in cell morphology and the subcellular localization of specific proteins of interest in large numbers of cells in response to a particular stimulus. This is accomplished by first stimulating the T cells with the chemokines of interest. In the second step, the cells are fluorescently labeled and then analyzed by imaging flow cytometry. In the final step, the number of morphologically polarized lymphocytes and the amount of segregation of the fluorescent markers can be evaluated based on the location and the intensity of the pixels for each staining, depending on the chosen mask. Ultimately, this imaging flow cytometry technology can be used to rapidly and robustly quantify the morphological changes of lymphocytes after chemokine stimulation, as well as the location of specific proteins of interest within the polarized cells.
The main advantage of this technique over existing methods, like microscopy, is that a large number of cells can be rapidly processed and automatically analyzed, securing statistically strong analysis of the morphological changes and the protein distribution in chemokine-stimulated T lymphocytes.
For chemokine stimulation of the T cells, begin by washing 5 × 10⁵ cells per experimental condition in warm HBSS supplemented with 10 millimolar HEPs. Centrifuge for 5 minutes at 340 × G at room temperature. Remove the supernatant and resuspend the pellets in 0.3 milliliters of warm HBSS HEPs per tube.